Welcome. Okay, so today, if you didn't know, at this far end of the conference in this tiny corner over here that you're all crammed into this room, we're going to be talking about the microbial ecology of CF. So I'm Spencer. I'm from Alabama, which was easy to find out. This is Paul. He's from UPenn. Okay. So the objectives of this particular workshop are rather broad, but I think that's a good thing. So a lot has changed in the past few years on how we think about infection, certainly in CF with different hard to treat organisms, but we still have these highly effective modulators that have shown up. Yet we're still treating our patients. We're still having to understand how these microbes and pathogens work because disease is still persisting. But it's a lot different than it used to be. And there's still hard to treat organisms, and there's still big gaps of knowledge that we need to figure out. So this particular workshop is going to try to touch on a lot of that. We kind of organized it around looking at some specific hard to treat organisms, some active, pretty new research that's coming out on those. I'll be honest, we tried to minimize Pseudomonas a little bit, but it's a little impossible, okay? Partially because there's 2 talks on Pseudomonas out of 5. Full disclosure, Paul definitely works with Pseudomonas, and I do a little bit. So we have some wonderful talks, some upcoming CF celebrities for sure. Dr. Nguyen from McGill, she's here. This is her second talk of the conference, by the way. She's going to talk a little bit about Pseudomonas as well, and I'll let them do more of their introductions. Dr. Morgan is from Washington. She's going to talk a little bit about Aspergillus and ETI effects, so this will be quite interesting as well. Dr. Hillam from Dartmouth is going to talk a little bit about microbial diversity and pathogens similar to responses of highly effective modulators. Elizabeth, Dr. Bergner is here. This is her second talk of the morning. Since I think many people were in the exact same session before and everybody migrated to the same spot. So that's a pretty big accomplishment in my opinion. So that's pretty nice. She's going to be talking again. I think they may ask Luke to have her do the plenary later too, since she's just been talking all morning. So things will be great. And then to round us out, we have Dr. Corley from National Jewish, who's going to talk a little bit about Mycobacterium abscessus. And so we'll get a good blend of the ecology in the CF airway. Okay, housekeeping. Paul and Spencer's workshop rules. As you found out, interactions, compliments, and questions are strongly encouraged. Wait to the end, okay? These presenters worked very hard on these presentations. There are CF celebrities and bigwigs in this room. Please come introduce yourself to them and congratulate them in person at the end. They did a lot of work into this, okay? There will be time for questions at the end of each presentation. And I think this is the only workshop that is chaired by 2 pediatricians in the whole conference. So brace yourselves and buckle up, okay? And there are a few pediatricians in the room, so I see some. So, okay, first up, Dr. Nguyen. Thank y'all.
So, I'm going to start.
Okay. Let me just make sure I know how to start.
When the beeper goes off, you have to stop.
Okay.
If there's no beeper.
And then I jump. Okay. Well, thank you very much, Paul and Spencer, for the invitation. I am buckling up, and this sounds like an awesome collection of talks. So it's my honor to talk to you today what I hope you will not find to be heretical, which is the idea that Pseudomonas might be intracellular, sometimes at least. Okay, can I make this work? Next. I know how to operate this.
I use the mouse.
You used a mouse, okay. Okay, so I have no disclosure relevant to this talk. So all of you know Pseudomonas as an extracellular pathogen, but what I'd like to convince you today is that maybe it does have a lifestyle that most people do not think about, and perhaps it is relevant as a bacterial reservoir for persistent infection. And so it's been actually decades now that several groups have been observing intracellular Pseudomonas, primarily in a variety of different non-immune epithelial cells, bladder, corneal, and even bronchial epithelial cells. And most of that work has been in vitro. And there's also some animal infection model, chronic bladder UTI infection or chronic corneal infection, where people looking at those mouse tissues can also document intracellular Pseudomonas. And this is not entirely unusual, as there are a number of other conventionally intracellular pathogens like Burkholderia or staph that have also been associated with chronic infection when they can take on an intracellular lifestyle. And so we've worked with this in vitro cell culture models where we're in particular looking at some very low infection inoculum and able to see intracellular bacteria either by microscopy or electron microscopy. And part of the interest in that has been to see that clinical isolates of Pseudomonas, so CF clinical isolates, show a very high diversity in their ability to survive intracellularly. And most bacteria, most isolates, I may say, may not survive at great bacterial burden over time, but you do find some clinical isolates that actually look like they could replicate with, maintaining a high or increasing bacterial over time. And if you're interested in that in vitro work, we have poster 78, where we've looked at a collection of clinical isolates, and there's kind of an inverse relationship between the ability of bacteria to survive intracellularly and their cytotoxicity. And this is very much driven by whether or not strains are type 3 secretion proficient or deficient. And we can demonstrate that the loss of type 3 secretion, which you see in a lot of chronic CF isolates, actually will promote intracellular survival. But that's not what I'm here to talk to you today. But that in vitro work has really led us to one kind of fundamental question, which is that, yes, these are in vitro observations, but is there actually evidence that this happens or can happen in human tissue, in particular human CF lung tissue? So a few years ago, working with some colleagues where they are doing lung transplant just down the street from us, there was a very interesting case study, which I'm going to present you today. And that case study came from a patient that was known historically to be chronically infected with Pseudomonas. But about 2 years prior to lung transplantation, their sputum culture started turning not growing Pseudomonas anymore. And repeatedly over about, kind of 20-something sputum cultures, there was only one incidence where we could find Pseudomonas. And the rest of the time, it was really just Stenotrophomonas maltophilia as the dominant organism and a variety of other kind of, probably other colonizers. And then in June of 2017, the patient underwent a lung transplantation, and then we sampled this fresh tissue. And when we sample this fresh tissue, what we do is we take the tissue, take what we will call pre-lysis tissue. So this is kind of all the extracellular secretions, what might be outside of intact cells. We do some CFU count, and then we treat the tissue with a combination of tobramycin and colistin. This was driven by the MIC susceptibility of the cultures available at the time, which the idea is to kill what is accessible extracellular bacteria. We control that this is sterile, and then we lyse the tissue, and then we plate this post-lysis, and then what do we find? And so first, in all the extracellular pre-lysis tissue, we were able to find Stenotrophomonas, which is what was expected from the pre-transplantation culture. But what was surprising is that once the tissues were lysed, that we could find Pseudomonas. And now, this is not necessarily, intracellular, but this was the first kind of indication that where was the Pseudomonas hiding out, and hiding out even, to the extent that it could not be killed by your, antibiotics, and then was required lysis. And so with this tissue, we then wanted to try to look for Pseudomonas by microscopy. And so we started developing some methods, first just by immune immunohistochemistry. Now, there are, if you go back to the literature, there are some certainly older studies that have looked at autopsies or explant tissues doing immunofluorescence or immunohistochemistry. And it had never really been noted that there were bacteria within the airway epithelial cells. And in part, going back to that literature, it's like, well, I think, people were not specifically looking for it. It's kind of looking for a needle in a haystack. And you really get overwhelmed by the amount of bacteria in the lumen, and that's predominantly where it is. So we wanted to kind of revisit some of that methodology. We compared a couple of different antibodies available, and that does matter because we're working with clinical isolates. So antibodies, depending on how they're raised, may not be able to detect what you want. And then we did a variety of controls, which I don't have the time to show you here, but I'm happy to talk about, where we wanted to make sure the antibodies were specific and sensitive enough for detection. And so first, we were able to see, as expected, some bacteria in the lumen, although that is relatively minimal because of the processing. All these tissues are washed first before they're fixed. And, these bacteria can be seen in cells like neutrophils or in aggregates in the sputum. And then we went and searched for intracellular bacteria, really honing in primarily around airways. And so this is where, if you look, we started being able to see every so often some, intracellular signals that in our eyes were compatible with bacteria. Now, what you can see is that, they were really just punctate, small signals. These were not large aggregates of bacteria. And, so this was in the patient in whom we were not able to detect Pseudomonas extracellularly. So this was really quite intriguing. And so from there, this is N of 1. It's like, well, can you find it elsewhere? Is this like the, unique case control? And so what about, can we go find it in other explants? So then we proceeded to go start collecting lungs from additional patient undergoing transplantation. And we developed this workflow where we would simultaneously do some culture-based detection and then fix them to do repeated immunohistochemistry. And then in a subset of them, attempted to do a novel imaging with a technique called microbe detection after passive clarity technique, MIPACT. And so over the course of a couple of years, we were able to prospectively collect a total of 7 lung explants. And here I'm showing you kind of just the microbiology of the culture. And what you see is unfortunately for us, there were no other Pseudomonas culture negative explants so all of these patients, what you see here in the black dots are the Pseudomonas positive cultures, and in the white dots were non-Pseudomonas. So everybody else had Pseudomonas over the course of 2 years or so prior to their transplantations. Sometimes the Pseudomonas was not always detected in the, for example, CF6 here did not have Pseudomonas in the year prior to transplantation, but you had some before. And at the time of transplantation, many of them, their sputum, because they're getting so many antibiotics, were often culture negative for Pseudomonas. When we analyzed the tissue pre-lysis, we could certainly find, though, that the Pseudomonas was still there, and then post-lysis there were. So with those, that was just a culture confirmation of the status of Pseudomonas. And then when we took these tissue, what we set out to do first was to do bright field microscopy. And so in these tissues, we first looked at immunohistochemistry, as I shown you. And then we went ahead and also did some morphometric analysis of airways to try to just characterize where are these intracellular bacteria. And relative to the various airways we were able to analyze. And to do some histopathology analysis to try to see if was there any correlation between the detection of intracellular bacteria and the degree of inflammation and damage. So I'm going to show you a few of those findings here in that cohort of patients. And also we did some controls with non-CF lung tissue explants. And so here, this is just a description of how commonly did we find intracellular bacteria when we sampled each lung. Each lung was sampled between anywhere from about 3 to 6 different samples. So it was not an extensive, but it was not neither a unique. We tried to randomly go in multiple lobes of each lung. And so we were able to find this in 3 out of the 7 lung explants that were detected. And these were seen in a variable number of bacteria. And where were those bacteria? This is where we start characterizing the size of the airway. And so here, the data is just displayed by patients, and all the red dots are the individual airways where we detected intracellular bacteria, Pseudomonas. And so in the 3 patients that did have some Pseudomonas, we were able to find this in a range of small to large airways. So this is the perimeter of the airways. And if we looked at this from the perspective of comparing airways that did or did not have Pseudomonas, what we found was that the airways that had intracellular Pseudomonas tended to be slightly larger in perimeter. So all things being equal, you more commonly had intracellular Pseudomonas in slightly larger airway. And if you broke those down in terms of the size of the airways, whether they were less than 1 millimeter in diameter, 1 to 5, or larger airways over greater than 5 millimeter, then there was a trend towards larger airways having greater number of cells containing intracellular bacteria. Now, how does this relate to potentially extracellular bacteria? So the proxy that we had was just simply looking at that luminal Pseudomonas signal. So now remember that the luminal signal was relatively modest to the extent that, so it's an imperfect measure because there are some washing steps that goes into the processing of the sample. But nonetheless, there was a trend where the airways that had intracellular bacteria tended to also have more intraluminal Pseudomonas signal. So generally, probably a more infected area compared to the ones where no Pseudomonas was seen. And if we looked at now a scoring of pathology, and this was our pathologist looked at this and in a semi-quantitative manner scored whether there was lung damage and lung inflammation, there was no statistical difference that we could detect in the limited samples that we had. Now, so those were kind of the bright field microscopy analysis that we did with immunohistochemistry. Next, we wanted to try to find another independent matter in which to find these intracellular Pseudomonas. And we collaborated with Peter Jorth and Diane Newman, who had in her lab developed this technique called MyPact. And essentially what it is that we took these samples of the lung, fixed them, embedded them in a hydrogel, and then delipidified that so that it becomes optically clear. And the purpose of this process is that one can look at thick sections in a matter and then combine that with various staining or immunofluorescence so that you could recognize at a larger scale with some airway structures and alveolar structure. And the goal of this was to go hunting for, again, microscopy, another method for microscopy detection of Pseudomonas. And here we used the antibody for Pseudomonas as a way to localize Pseudomonas. So I'm showing you here some pictures. This is, for example, a very large airway, one of that we were able to see. I'm showing you the picture because it's particularly pretty, and you can clearly recognize what the where the airway structures are. You can see the nuclear stain, and the WGA just gives you a sense of what the overall three-dimensional structure are. And then in the bronchoepithelial layer, you can identify some cells which, with the 3D reconstruction, we think that the signal is intracellular. And when we tried to apply some markers to specifically locate the epithelial cells here with the E-cadherin antibody, then you can also see areas where you see a Pseudomonas signal that in a three-dimensional reconstruction appears to be intracellular. So with that, where are we at? Well, I think that this is the very first demonstration that in human CF lung tissue, we have, I think, evidence that is converging towards showing some intracellular Pseudomonas. And this, I think, kind of lends plausibility to the hypothesis that this could be a reservoir. Now, the study, certainly has some limitation. There's some sampling and processing bias. These are relatively infrequent events, although with the way that we have samples, I can't claim to be able to quantify this in any meaningful manner because I don't know what the denominator is. And this is also end-stage lung disease. I'd love to be able to say, well, this happens earlier, but I think, practically speaking, it's an impossible task to answer. So we have what we have with end-stage lung disease here of the lung explants. And so there are still, certainly many, many unknowns about, how common or what type of epithelial cells. Are there other cells that are involved? And hopefully some of the in vitro work would be able to partially answer those questions. And with that, I'd just like to acknowledge the people in my lab that have contributed to the data. I've shown you collaborators that have worked with us to provide access to the lung explant, the pathologist, and the imaging that we have done. And with that, I'd be happy to take some questions. Thank you.
Please, yeah.
Come on up.
Beautiful talk. Microbiology question. I'm wondering if the intracellular pool has arisen from the endogenous extracellular pool, or it's a new strain. And any kind of morphotypes, are they mucoid SEVs?
So we have so far not been able to identify any phenotype that distinguishes the intracellular from the extracellular clones that were isolated. We have not done any sequencing to look at clonality at this point, so I can't tell you that. But that would, it certainly begs the question in terms of the corresponding, at least in at the local level, whether they're related or not.
Thank you. Great talk. So I think I talked to you about this last year that my name is Doe from UAB. I work with a rabbit sinusitis Pseudomonas, and I noticed that a lot of Pseudomonas in this tissue. That's why I said, oh my God, topical antibiotic may not work. And I have to learn about the rabbits a lot or fluorescent. I'm going to ask you about later about the Pseudomonas staining. That's very tricky. My question is, have you ever DCF patient an antibiotic? The reason is that I showed in my patient grant rally yesterday, when patient on antibiotic rinse, these are larger airways, and then Pseudomonas appears to be hide in the tissue. Is that something you notice? Because when you put on these patients a topical, a lot of antibiotic regimens, that could look like Pseudomonas appears to be going into the tissue instead of the way they to survive. And do you notice any protease could be a role for this, for intracellular invasion of the Pseudomonas?
So we have not tested specifically any protease deficient Pseudomonas in vitro in terms of looking at whether or not it affects the invasion process. But what we have tested, and this is because my lab has a long-standing interest in LASAR mutants. So indirectly, a lot of these LASAR mutants are deficient in a variety of proteases, and we did not see any difference in vitro. Now, having said that, what are the processes that allow Pseudomonas to invade, and can we recapitulate some of that, or the most important ones in vitro? To me, I'm not sure. Certainly, I think one of the things that we observe in vitro is the integrity of the epithelial layer will change the rate of invasion. And so if you have, if somebody's sloppy and they're letting their layer being too confluent, or if you cause, quote unquote, what it would be an injury to the epithelial barrier, you will have increased invasion. So with that, and we don't model that and we don't measure that, but what you could imagine is if you do have epithelial barrier injury and proteases potentially being important to that, that could be a port of entry that will promote the internalization of Pseudomonas, because and certainly in stratified epithelium, whether you're the basolateral or apical, that matters.
I have a quick question. I want to know, Dr. Wynn, your thoughts on this large airway aspect. Because when I saw that, my gut feeling was like, oh, these are big bronchiectatic bad airways that you're seeing. But it sounds like your block analysis and that they didn't really look like that. And so I wanted to, because I mean, I think that, that would be really cool. And that's like the first thing that my gut thinks is going on, but I wanted to hear your thoughts as you guys have dug deep into this.
Yeah. So the image that I show it because it is, very, very pretty. Now, it is an outlier of, what we're able to see. And so, I think that what is more accurate in terms of what is the distribution of airways that we were able to analyze, there were not many large airways. So that's just kind of, putting it out there. And part of that was just our sampling. We were trying to be kind of agnostic to the degree of damage, because one is, this is done at the fresh out of the surgery, and so you look at it from the outside, you don't look at it from the inside, and we were just kind of going and punching holes and hoping to get a smattering. And so when we described this over the range of small to large airways, we were sampling more kind of the small and medium, and there were not that many large airways.
Hi, Paula Zamora from Dartmouth. So maybe from your in vitro modeling, do you know, like if you need the bugs to be alive, like if you were going to fix the bacteria and then add it into your epithelial cells, like is it a mechanism that the bugs are kind of like telling the epithelium to like bring them in, or is it the epithelium that can just like bring stuff in, like?
Is it a passive or active? Yeah.
And then can I ask?
Yeah.
So then when it goes inside, do you know if it's like surrounded by like a double membrane vesicle, and then does it fuse with the lysosome in some point, and when? Okay.
Yeah.
Sorry, too many questions.
No, I think they're great questions. So to your first question, I don't know. We've never tried to do this with dead but physically intact bacteria to see if there's a passive kind of engulfment process. So I have no idea. To your second question, where is it located? And I think that is still something that we're working on, because the different people have observed different things, and this is partly the challenge with the in vitro model is, well, which cell type and the cell type certainly does seem to matter. Which MOI? So, are you doing an infection at 100 bacteria to 1 versus, and so we have chosen to do these infection at a very low MOI, in part because it is the only way to preserve enough viability of the host cells and to maintain. And so we do these in vitro experiments up to 5 days long, and the epithelial cells remains viable, but you only have one bacteria per epithelial cell. And that MOI does appear to affect the trafficking and where the bacteria ends up. So some people have described bacteria in the cytosol, kind of just like hanging out there, and others in single membrane, and others in double membrane, and sometimes in multiple types of vesicles, depending on what the bacteria is expressing. So I think that there are many different paths, and it remains to be seen which one matters and which one are driven by the host and which one are driven by the bacteria.
Great. Thank you very much. Beautiful talk.
Next up is Dr. Morgan.
How do we get out of here?
Maybe the X.
Nope.
Oh, there we go.
Right.
Oh, good. It worked. Well, thank you for joining us. My name is Sarah Morgan, and I'm a research scientist in the Sing Lab. And full disclosure, based on the introduction to this talk, I'm actually a Pseudomonas microbiologist masquerading as a fungal biologist today. So I guess that's how you get talks now. But anyway, so today I want to talk with you about some of our really exciting work looking at the filamentous fungi Aspergillus fumigatus and its response to ETI. If I can get the slides to advance.
No, that's better.
There we go. Okay. There it is, Pseudomonas. So, as we all know by now, ETI is a very remarkable treatment. It greatly improves lung function for many people. And it also reduces, but does not eliminate infection of many of the bacterial pathogens. And so this is some data from our recent publication looking at looking at bacterial pathogens in the CF lung, pre and post ETI. And as you can see, the total abundance of Pseudomonas drops by about 2 logs, the same for Staph aureus. But only about a third of the patients in this study, or the participants in this study with Pseudomonas aeruginosa, were able to clear the infection enough that we had repeatedly negative cultures, and only about a quarter of the participants who came in with staph had repeatedly negative cultures. And these numbers get even worse if you look at this by the genome. So by ddPCR, we only saw about 11% of Pseudomonas subjects negative post-ETI that came in positive pre-ETI. So we know that bacterial infections are still a big issue in the post-modulator era. And one of the questions that keeps coming up is, what about other pathogens? And so today we're going to look at Aspergillus fumigatus. So Aspergillus fumigatus, which I'm going to just call AF because it's a long name, is the most common fungal pathogen in CF. It is associated with reduced lung function, and it's difficult to treat. It's particularly difficult to treat in the post-modulator era because it's generally treated with triazoles, which inhibit the CYP3A enzyme that's cleared by ETI. So the two treatments don't work well together. And so it's very important to understand in the post-modulator era what is going on with AF. And there was a really great study that came out last year looking at the prevalence of AF after ETI. And so here it's a single center retrospective study, and they found that before ETI they had a much higher prevalence of Aspergillus than afterwards. You can see each dot's here a patient, and they only had a few patients that were positive. So this is a really intriguing result. And if this finding holds true across the CF population, it could be very important for the health and treatment of people with CF. So therefore, we wanted to look at Aspergillus positivity in the prospective multi-center study, PROMISE, which we've been involved in for quite some time now. So PROMISE prospectively collected sputum pre and post ETI. They had 236 individuals enrolled at 27 sites across the United States. It was focused on understanding how pathogens change in response to ETI, but due to a combination of cost and low sputum volumes, fungal cultures were not included in the original study. However, sputum was saved and frozen for DNA extraction and fungal study later. So in order to study this, we developed a droplet digital PCR assay, which quantifies Aspergillus genomes in the sputum. Droplet digital PCR uses a reaction setup similar to qPCR, where you have 2 primers and a probe used to detect the template, but then ddPCR splits this reaction into 18 to 20,000 droplets, each of which is individually assayed. And this gives us much better quantitation and much higher sensitivity than traditional qPCR. It also allows us to use small sputum volumes, which is important in the post-modulator era. and it's less prone to PCR inhibitors, which are a problem both in fungal cultures and in CF sputum. So this was a good assay for us to use. We selected 2 targets. So the first one is the BEN-A gene, which encodes beta-tubulin. And one of the major advantages of this is that it's a single copy per cell. So whatever we get for BEN-A, we know represents the number of chromosomes in our sample. It's also highly divergent in other Aspergillus species, which allows us specificity. However, in cases of very low abundance, it may be difficult to detect BEN-A. So to get a little bit better sensitivity, we also use the ITS sequence. So we're using here ITS1 and ITS2 on either side of the 5.8S ribosomal DNA. And these sequences are commonly used for identification of fungi. And so we selected Aspergillus fumigatus specific sequences. So we are mostly looking at Aspergillus fumigatus here. We did occasionally see, if we had very high levels of other Aspergillus species, a low level of false positivity. So, but those conditions are likely rare in CF sputum based off of the literature. I'm going to talk very briefly about assay development. If you are really excited about assay development, I'm happy to answer questions, or you can come see my poster. But I just want to show a couple of things to hopefully convince you that this assay works before I show you the data. So because one of our primary goals of this study was to measure quantitative changes, and we thought that quantitative changes could be very important, even if participants don't clear the infection. We wanted to know how accurate quantitation of this ddPCR assay was. So we just simply made dilutions of the spores. And so you can see that here in black, in the black line, those are the spores added. And then we did replicate extractions and quantitated it. And as you can see, as you would expect, the BEN-A quantitation very closely follows the amount of spores added. And the ITS is very nicely linear, but it's shifted up about 37 times due to that higher copy number of ITS. The other big concern that we had was limit of detection. Because we were expecting the potential for very low numbers, we wanted to know how low can our assay measure? How sensitive is this assay? And so again, we're just gonna take serial dilutions of spores. And this time we took 10 replicate dilutions, and we assayed each one just simply for positive or negative. Did we see it? Didn't we see it? And as you can see for BEN-A, we were able to detect the spores at about 2,500 spores per mil, which is not as low as we would like it to go. But for ITS, we were able to reproducibly detect it down to about 50 spores per mil. And this is on par with what we would expect from most clinical culture methods. Okay, so I've talked with you a little bit about the ddPCR assay that we used. It's accurate and it can go relatively low in quantitation. So let's move on to the big question that we had. How does ETI affect Aspergillus infections in people with CF? And we're going to use this PROMISE study cohort that I introduced very briefly early. Just for anyone who's interested in what this cohort was, we tried to get a very good cross-section. And so its age ranges 12 to 62 years. The FEV also ranged from about 29 to 130% predicted. And participants were included irregardless of their infection status. So we had no knowledge going in what their prior infection status was. Due to low sputum availability and some of the sputum already being used for culture, we were only able to study 114 of the participants for our Aspergillus study. And we were looking for participants that had a culture or had sputum available before and either at 1 or 2 years post-ETI. 86 of these participants also had a sputum available at 1 month, and several of them had sputum available at other time points as well. Okay, so the first question that we want to look at is how does Aspergillus change in prevalence across this whole 114 participant cohort? And so here I'm showing you the prevalence. And if you follow the blue line, pre-ETI, our prevalence was about 28%, which is smack in the middle of what we would expect based off of a lot of the literature out there on AF prevalence in CF. And within a single month on ETI, this had dropped down to 5%. So a dramatic drop in the frequency that we were able to detect Aspergillus, and it stayed low out at 1 year post-ETI. You can see the same trend with the BEN-A, but due to our lower limit of our higher limit of detection, we weren't able to see this in as many participants. So I'm going to focus in on the 1 month time point because that's really where all the action seems to have occurred in this population, and look at the quantitation. So here's the quantitation for BEN-A. We had 13 participants come in positive for BEN-A pre-ETI, and after a single month on ETI, we were only able to detect that in 1 participant. And the rest you can see dropped as much as 3 logs in abundance. Similar story in ITS. Here we were able to detect it in 25 of our participants, and within a single month on ETI, this dropped down to only 3 participants being positive. So again, within that single month, a large drop. And here you can see we were able to detect as high as 10 to the 6th copies of ITS pre-ETI. And even a lot of those participants with very high AF levels pre had very low levels post. So now I want to just show you the time course for what happens to all of these participants. And because our BEN-A detection is a little bit lower, we're going to focus on ITS for the rest of the talk. And I'm also going to start with the participants who became negative. So of our 32 participants that came in positive, 27 of them, we were never able to detect another Aspergillus positive sputum sample from them. And many of these participants had given us 4 post-ETI samples at that point. We had only a single participant, shown here in this green line, that had multiple positive samples for ITS after starting ETI. And this participant, you can see, also dropped about 3 logs in abundance and stayed with a much lower abundance throughout the rest of the course of the study. There were 5 additional participants that were positive at baseline and had any positive samples post-ETI, so they each had a single positive sample post-ETI, and the rest of their samples were negative. So it does look like most of the participants that came in with Aspergillus, we were no longer able to detect that Aspergillus post-ETI. So some of you might be wondering, what about the other participants in the study? We did follow them as well. So of the 83 participants that came in Aspergillus negative, we only were able to detect Aspergillus post-ETI in 2 of them. So it's not clear at this point if those are potentials for new Aspergillus or if maybe we just missed it at the pre-ETI. But it does look like, in any case, acquisition of Aspergillus is relatively rare in this population as well. So I do want to say outright that this study has a lot of limitations. We are not making any claims at all that Aspergillus is no longer a problem in CF. It's still definitely a problem. There's still definitely some participants, some patients who will have Aspergillus. And because of our ddPCR assays, if it's at very low levels, we probably didn't see it. And due to sputum volume limitations, we were only able to look at about half of our PROMISE cohort. And it's also really unclear how this decrease in AF density is going to affect the lung disease, because there's so many other factors that are also helping to improve these patients' lung disease. And this study only goes out to 2 years, and it'll be very exciting to see what happens after that. We've enrolled patients out to 4 years now, so we will keep watching them. So in conclusion, our ddPCR assay has comparable sensitivity to standard culture and is able to quantitatively measure AF. We saw that most participants, about 84%, came in positive, became negative. In comparison, again, going back to PA, that's compared to 11% in Pseudomonas, so quite a difference that we're seeing there. And almost all of our baseline negative participants remain negative over 2 years. And with that, I would just like to thank Pradeep and everyone in our lab, and Dave Nichols and Gina Hong for helping us get excited about Aspergillus, and Robert Kramer for answering all of my questions as I was figuring this out. And of course, to the Promise Study group as well. Thank you.
While Ron is coming up, I'll ask you a question.
Okay.
Maybe I just missed this, but I know that that cultures were not done for Aspergillus in the original study. Do you have any clinical correlation from the sort of standard culture that corroborates this? I mean, how many of those who are ddPCR positive have a clinical culture of Aspergillus?
Yeah, that's a great question. And I actually cut that slide for time. So we actually took a bunch of sputum samples that had been cultured at the UW clinic for Aspergillus and took the remaining sputum from that culture and assayed it. And we were able to find that, so our clinical lab does a semi-quantitative streak. And so they report the number of colonies that they see, although it's, not exact CFUs. And if they were able to see more than 2 colonies, we were able to detect it every time. If they were only able to see 1 to 2 colonies, we were able to detect it about 80% of the time. So pretty close, I think.
Yeah.
So I'm Ron Rubenstein from WashU in St. Louis. Do you have any data on whether these folks had a diagnosis of ABPA? And do you have serial IgEs on these folks, which are collected in a registry, that you could probably go back and get to see if there's any change? And also, were these folks treated with antifungals before you got them?
Yeah, those are all great questions. Unfortunately, we do not have any of the data about them before they entered the study. And we don't have any of the other things that you asked for either, but those would be great to have.
My only
yeah.
comment, Ron, is that somebody sitting at this table may have funding to do that here in a second. So,
Yeah, so-
and works with Sarah to do some of these promise samples to look at exactly some of the things you were asking.
Yeah.
Yeah, that's going to be really good context for these data.
Yeah.
To be interpreted.
Yeah.
Yeah, that's the critical next step for this data to really be interpretable.
Katie.
Katie Heiser from National Jewish Health. Ron took my question, but I'm still here. So I just want to say lovely talk, but also just to add that as a clinician, we often don't know what to do with Aspergillus in the sputum. Do we treat it? Do we not treat it? If it's not floridly ABPA, is it causing problems with other pathogens? So I think that following this cohort and also following what else is going on with them could actually be very informative moving forward.
Thank you.
Hi, very interesting. So do you know if the ETI has direct effects on the Aspergillus, like antifungal?
So Rob Kramer's lab actually has recently published on that, and they did find some antifungal effects. And I believe they have a poster here today as well, but I don't know that poster number. I'm sorry. Well,
Okay. Thank you, Dr.
Thank you.
Morgan. Thank you.
I think so. Okay.
thank you to Spencer and Paul for the introduction and reminding me that I need to put in shorter talk titles when I submit for talks in the future. So I'm going to talk to you today about bacterial pathogens persisting in the upper respiratory tract following the initiation of ETI therapy. So I'm in the Bomberger lab at Dartmouth College. Should say I don't have any disclosures related to this presentation. So for anyone not familiar with our lab, we have a long-standing interest in sinus disease. Here are a few of the papers that we've published recently, interested in both the host and pathogen side of sinus disease in cystic fibrosis. And the reason we're so interested in this is that chronic rhinosinusitis is a really common comorbidity in people with CF. So you can see from this plot here from the CFF patient registry that by the age of about 30, about 50% of people with CF also have chronic rhinosinusitis. And this causes chronic inflammation and development of polyps in the sinuses, which cause sort of real bad quality of life symptoms such as loss of sense of smell, buildup of pain and pressure in the face, and postnasal drip, which can have a knock-on effect on sort of nutritional symptoms as well. But as well as these quality of life issues, the sinuses are also pose a risk to people with CF in terms of their health and infection. So there's a body of literature that indicates that bacteria from the environment first colonize the sinuses before then traveling into the lower airway to cause lung infection. And so this could be, targeting the sinus could be an option for sort of early eradication therapy, as well as being a really important consideration for those people being considered for lung transplant if those bacteria sort of hang out in the sinuses and then seed the lungs that people have transplanted. So I hope I can convince you today to consider the sinuses as more of a CF disease state in their own right. So talking now about our study that we have, it's an observational longitudinal study completed in 2021 of 38 adults with CF at the UPMC Adult CF Center in Pittsburgh. These were collected by Dr. Stella Lee. So we collected paired upper and lower respiratory samples. So the upper respiratory samples are taken endoscopically with a sheath and a swab into the right maxillary sinus, and the lower airway samples are either a sputum sample or a throat swab. And the idea of taking this sort of big biobank of samples was to look at a whole range of things, both on the host and pathogen side. So sort of microbiome, Pseudomonas aeruginosa, in particular genomic sequencing, host inflammation, and also viral pathogens that are in there as well. But what I am mostly going to talk to you about today is the 16S microbiome analysis, looking at the pre and post ETI microbiome in the sinuses in the upper airways of people with cystic fibrosis. So we managed to, I think you can see this okay, we managed to capture the initiation of ETI treatment in 14 people with CF. The samples of the people that we captured that for are highlighted in red here. So that's a total of 140 samples pre and post ETI. And the question that I wanted to answer was, does the upper airway microbiome change in these people post ETI? So the first thing we looked at was the sort of total bacterial abundance in the sinuses following ETI. So we carried out a 16S qPCR to generate copy number totals per sample. And here what I'm showing you is that the colors represent each person in the study with at least one pre and post ETI sinus sample. And then the points are the mean 16S copy number for that person for all of their samples, pre or post ETI. And we can see that most people experience a drop in total bacterial abundance after starting ETI treatment. And although this wasn't shown to be statistically significant, it is consistent with studies carried out in the lower airway, particularly the PROMISE study that we just heard about. So if I then zoom in a little bit deeper into the microbiome data itself, I used this Maslin R package, which uses multivariable associations and linear models, specifically for microbiome data, to identify taxa that have significantly different abundance in whatever conditions you tell it, in my case, pre and post ETI. Pseudomonas was identified as the only significantly differently abundant organism in these conditions. And I think this difference in relative abundance is pretty striking. We go from, anywhere up to sort of 100% nearly 100% Pseudomonas in these samples, down to like well below 1%. But I think what I really want to highlight here is that although we have this massive drop, the Pseudomonas is not eradicated. There's still sort of this just below 1% detectable Pseudomonas hanging out in the sinuses after ETI. And I'm not going to dig too much into this today because my colleague Catherine Armbruster will be talking about this tomorrow at 9:30. It's a great talk. It's going to be about the persistence and evolution of Pseudomonas in the sinuses following ETI. So go and check that out if you're interested in hearing more about Pseudomonas. But I'm sure that Paul and Spencer will be glad to hear that I'm not going to talk about Pseudomonas for the rest of this talk. So I'll zoom back out a little again here. And so I wanted to look at the last available pre-ETI sample for patients, which here is in gray, and then all of their post-ETI samples, and along the x-axis I have months from their last pre-ETI sample. And so I made these type of plots for every taxa that we identified, and then kind of picked out the ones that were either genera of interest or had something interesting happening pre and post ETI. A lot of the samples were very low abundance and remained at low abundance. But what I want to draw your attention to here is the kind of striking difference in the way Staphylococcus seems to change post ETI compared to the other genera that I'm showing you here. So the dots connected by lines here are all from the same patient. So you can see we get this massive increase in Staphylococcus relative abundance, and then it seems to be maintained at really high levels for the months following ETI. So my question here is that these changes are in relative abundance, obviously. So it was really important for us to look at what changes in the microbiome in terms of the total bacterial abundance and the relative abundance of other taxa. What changed in that to determine this huge increase in relative abundance in Staphylococcus post-ETI? So what I'm showing you here is 16S copy number per sample along the x-axis, and then the relative abundance of each taxa in that sample on the y-axis, and then the different taxa are colored in different colors. So if you look up at the top of the plot here, you can see that the samples generally tend to be dominated by Staphylococcus and Pseudomonas, but there's a little bit of Escherichia and Achromobacter in there as well, and dominating these samples. And if we look in the bottom left corner, we can also see that Staphylococcus is present but not dominant in some of these sort of lower bacterial load samples. So then if we look at the post-ETI, we can see that that variety in the dominant organisms seems to be almost entirely gone post-ETI. And if we look just at Staphylococcus, you can see that it is now largely the dominant organism in the sinus samples post-ETI. So my next question was, we know that Staphylococci can exist as part of a healthy nasal microbiome. So we wanted to determine if this increase in Staphylococcus abundance was perhaps due to inhalation of those nasal microbiota into the sinus, and then this establishment of those perhaps commensals into the sinus, or whether or not we're seeing dominance of Staphylococcus aureus and maybe MRSA in the sinuses following ETI. So the way we looked at this was using a microfluidics device. This allows you to multiplex up to 48 primer sets with your samples and then sequence those amplicons. So in our databank, we have 16S amplicon sequencing, ITS sequencing, which I won't talk about today, and then species and strain-specific primers for both Staphylococcus and Pseudomonas to help us identify which bacteria are actually in there, because obviously that's one of the major limitations of 16S sequencing. And then, so what I'll be showing you is basically yes or no binary answers for what we think we have in these samples, and this is determined by a 1% cutoff of the total counts for each amplicon in that sample. So just to give some context to the results I'm going to show, this is the estimated Staphylococcus abundance per sample. So this is not relative abundance. This is how, based on 16S qPCR and the 16S copy numbers, how much Staphylococcus we estimate to be in each sample, and each of these bars is from a different sample. This just indicates which patient each of the samples came from, so you can see we've got a real variety of different abundances across the patients. And then this is the amplicon data here. So in gold, I'm showing you the Staphylococcus aureus, blue is Staphylococcus epidermidis, and in red is the samples that we identified as MRSA. And so you can see that with these samples, we're getting a really high persistence of high levels of MRSA from pre-ETI into post-ETI in these patients in their sinuses. And so we're not seeing this establishment as we maybe theorized of sort of a pseudo-nasal microbiome in the sinus. We are seeing pathogens persisting post-ETI. And so to sort of wrap up what I've told you here, pre-ETI, we see a really high bacterial load and sort of very dominant organisms, which then post-ETI in the sinuses, we see lowered bacterial load in most patients and significantly decreased abundance of Pseudomonas. But alongside that, we see increases in Staphylococcus relative abundance and a real persistence of pathogenic MRSA in the airway post-ETI. And obviously this is of concern because the sinuses, as I've said, act as a reservoir for pathogens that can then be seeded into the lower airway where they may continue to cause infection or drive inflammation and damage in the lung tissue. And now that we don't receive as many sputum samples from patients post-ETI, these pathogens may go undetected. And so I'd really like to encourage people to think about using the sinuses as a place to monitor, both as its own, as a source of bacteria, but also as their own disease site in cystic fibrosis. And so as I mentioned, we have a really substantial biobank of samples. So we have plenty of ongoing work. We're going to be using viral panel data to hopefully elucidate the effect of the viral bacterial co-infections on the CF airway microbiome. We'll also use whole genome sequencing to hopefully track the movement of bacterial strains between the upper and lower airway. And as I mentioned, there's some really interesting work from Catherine coming tomorrow on the persistence and evolution of Pseudomonas in the upper airway post-ETI. So with that, I would like to thank all of my colleagues at Dartmouth and Pittsburgh and Stella Lee, who collected the samples, who is now at Brigham and Women's Hospital. And my poster is number 90. I will be there today if anyone wants to talk about this in more detail, and I am happy to answer any questions.
Very interesting talk. I have a question, especially in light of the first talk that we had. If you've looked at intracellular Staph aureus as a possible reservoir, because there are lots of studies that suggest that Staph aureus can be intracellular, particularly in nasal polyps.
Mm-hmm. Thank you. We have not. We're not a huge staph lab most of the time. I'm probably going to start digging into staph a little bit more in light of this. So that's definitely something we could consider as part of our like CF model work that we do. Thank you.
Hi, that was really great. Sarah Clark from University of Colorado. So I'm wondering if you guys are interested in looking at the isolates and this idea of seeding the lower airway, if you can do some sequencing, maybe of paired isolates that you get from the sinuses versus the lower airway. And we've also heard at this conference about people using the throat swabs as another kind of way to estimate what might be in the sputum. So maybe all three sites. I was just curious.
Yeah, that is it. That's pretty much exactly what we plan to do. We have some of that data already for Pseudomonas. I don't think either of us are presenting that at this conference, but we have that data, and I imagine that we'll probably start to work with it in staph too. But yeah, we have, as I say, those samples, the paired samples, were collected on the same day at the same clinic. So they are truly about as paired as you can get. So it'll be exciting to see what comes of that.
Hi, Barbara Karl from Münster, Germany. Very interesting. So I'm especially interested in Staph aureus. And so do you have any information about the colonization with MRSA before in the nose of your patients?
We don't have any nasal information. We do have all of the clinical micro data from this. It's one of those things that it's taking me a little while to sort through because it's all like hand typed in notes, so it's not coded particularly well. So it takes me a little while to sift through and get it out. But I think there is pretty good concurrence between the clinical micro data for MRSA and what we see in the sequencing data.
Okay, thank you.
I have a bit of a technical question.
Yep.
For your specific for Staph aureus and MRSA, the MRSA seems high to me. It seems unusually high. How are you detecting MRSA versus?
So we have an LDH primer for Staph aureus, and then a MEK-A, and then a GSEA for the Staph epidermidis. So if I saw MECA and GSEA, I called that epidermidis. I didn't call that aureus. So I do need to go back. It's one of those things, it's as specific as we could get.
Yeah.
I'm going to go back and validate it. And I think we're actually going to pull a bunch of these out of the freezer and take a look at them. But there is, from my memory of the clinical microdata, there was a lot of Staph aureus in these patients. Yeah, a lot of them were culturing Staph MRSA.
Because you worry about MECA being in epi.
Yes,
Right.
yeah.
So,
Hi. Bergener from CHLA. I'm wondering if you have clinical data on what inhaled antibiotics these patients are on. I'm wondering if maybe now that they're on ETI, you're giving them inhaled anti-pseudomonals, it's driving the pseudomonas down and making them for the...
We do have that data. I haven't. We have tons of data for this study, and it's just I just haven't compiled all of it into that yet. But yeah, we do have that information. It's something that I'll definitely look into.
Thank you.
thank you. Let's see, X out of it here. Yeah.
Good to go. All right. I'd like to start with thanking the organizers for including me today to present my work on filamentous phage of Pseudomonas. Sorry, it's Pseudomonas. But I'm really focusing on the phage part. That entangles cilia and impair mucociliary clearance in CF. And all of this work was done at my prior institution at Stanford, but I've more recently moved to Children's Hospital of Los Angeles. I have nothing to disclose. I do apologize for those of you that heard my talk in the last session. My intro is pretty much the same. I will go through it for the people that weren't there, but bear with me. There's some cool videos and pictures after that. Our patients with CF, they're born with structurally normal lungs, quickly develop thickened mucus, get exposed to bacteria, influx of immune cells, end up with a vicious cycle of inflammation and infection that we can't break. And we know that Pseudomonas is the most common organism cultured from CF sputum. It's a bad player. It forms biofilms. It's very resistant. And we know there's poor outcomes in those that have chronic infection. Biofilms are bacterial communities encased in polymers. Think about a dental plaque. This also happens in the airways of patients with CF. And biofilms confer adhesion, antibiotic tolerance, immune evasion. And we know that in Pseudomonas biofilms, bacteriophage genes are actually quite upregulated. So in this study, looking at Pseudomonas aeruginosa and biofilm growth in comparison to planktonic growth, see that the top 9 genes that are actually upregulated come from the PF bacteriophage. The CoA-B protein, the one that makes the outer coat of the phage, being the most upregulated at 83-fold. So what is PF phage? So it stands for Pseudomonas filamentous. It is a non-lytic phage. It's a long, skinny string of a virus. It's got this protein coat, some cap and tail proteins on the end. It's got a single-stranded DNA genome inside. It's kind of folded back on itself. And it recognizes the type 4 pilus on Pseudomonas. That's how it infects it. The viral genome integrates into the bacterial chromosome, so you can detect it in bacterial whole genome sequencing. It's a prophage. When it's there was some kind of stimulus that causes it to go to excise, go into the replicative form, and then Pseudomonas extrudes it. And so there's all these PF phage particles that are extruded, but it does not kill the Pseudomonas. So it is a pseudolysogenic replication cycle. It does so without lysing Pseudomonas. I'm going to pause, make the same comment. This is not a talk about phage therapy. This is a phage that is present in many clinical isolates of Pseudomonas, and it's actually a bad player. It's more of a virulence factor of Pseudomonas. PF phage can assemble polymers into liquid crystals. So calcite is a crystal. When you look at that image there, you can see the line going straight through. You rotate the crystal, light is refracted, which makes that line look like it has moved down. That is birefringence. If you look at a colony of Pseudomonas that does not have PF phage, have a polarizing lens, and you rotate it, you don't see any irregularities. But a PF positive or PF producing strain colony, you rotate that lens or the lens, and you can see that there's areas of irregularity. That's birefringence, indicating that there's crystalline structures there. We call it liquid crystal because it's not solid. And this organization happens through charge, those depletion attractions. The phage is very negatively charged. All those CoA-B proteins that make up that coat all have negative amino acids sticking out. So it's a very negative surface. And then all of the polymers that are in sputum and in biofilms are also negatively charged. So it's through that interaction. So PF phage, liquid crystalline biofilms, we know from work in vitro, are more viscous. They're more resistant to desiccation, inhibit phagocytosis, limit antibiotic diffusion. And really because of that viscosity, we hypothesized that PF phage biofilms would impair mucociliary clearance in the CF airway. So first we ask, does PF phage make liquid crystals in the CF airway? And we did this by looking at explants. So we had a particular subject that we had well characterized. We had many sputum samples that had very high levels of PF phage, including one the week of transplant. And so we were able to get sections of the explanted lungs. And so on the left there, it's just a section where we're looking basically in an airway, and you can see there's lots of immune cells. There's some debris. It's a mucus plug. The image on the right is in the same airway. It's a different microscope, so it's not exactly the same section, but on the same slide in the same airway, using a rotopol apparatus, which allows us to quantify quantifies the differences in the polarized light, and basically with some software allows us to visualize that. So the green is areas of birefringence. So we can see that we do have evidence of liquid crystal in the airways of a patient with Pseudomonas that produces PF phage. And when we looked in a lung explants from a patient that had Pseudomonas but no phage, we did not see this. Okay, so there's liquid crystal in the lung, but how does it affect the respiratory epithelium? So for this part, we used air-liquid interface culture. Basically took nasal scrapings from both patients with CF as well as healthy controls, expanded the basal cells, put them on the insert, let them grow, took off the liquid from the top, so you end up with the air-liquid interface. The cells differentiate. You end up with both ciliated and non-ciliated or secretory cells. And so then we put PF phage on top of these ALI inserts. And so here is a scanning EM image of one of our healthy control inserts or cultures without any phage, but you can see they're nice, healthy looking cilia. When you add PF phage on top, they all just kind of stick together and really kind of get glommed down. I don't know how well it projects, but you can actually see the little squiggles of phage on the ends of the cilia. And then this is on a insert with CF cells. But again, you can just see it's all kind of tacked down and aggregated, and you can imagine this might affect mucociliary clearance. So to answer that, again, with more ALI cultures, we applied phage to the cultures, and then we collected mucus, as well as looked just directly on the inserts for evidence of liquid crystal. And then we also added beads to the top of the ALI cultures, and using video microscopy, we're able to track the beads and actually measure mucociliary clearance. So this is a video of looking down on the ALI insert and rotating the polarizing lens that we have attached to our microscope. And you can see there's that area kind of in that X where it stays white. So that's birefringence. If there's no birefringence, the whole window goes black. And that's on our ALI inserts, we get these areas where the cilia are kind of coordinated and moving, and there's a lot of mucus in these little tracks, and that's on one of those areas where we see the birefringence or evidence of liquid crystal. When we put the beads on our cultures, so the left is looking at a PBS control, and you can see those beads are moving right along, just kind of getting swept away. But on the PF treated insert, you can see most of those beads are just kind of stuck. Some are moving, but not nearly as well as on the PBS control. And so there's just representative images. We did this on multiple inserts and multiple windows within each insert, in both wild type or healthy controls, and then in CF cells as well. So in that first graph, we're looking at the length traveled of the beads, and you can see in the wild type with PBS versus wild type treated with PF phage, you get a significant decrease in the length traveled by the beads. In the CF, it's also significant, but the magnitude is so it starts out so low that it's a little bit harder to appreciate. And then when we look at the actual velocity of how fast the beads move, you can see again in the healthy control or wild type cells, a little bit more variable than the length, but a significant decrease. And in the CF, again, it already starts so low that the difference wasn't significant. But what was really cool and interesting and not necessarily expected is when we added ETI, it didn't help in the patients with phage, or in the cells with phage. So again, CF and PBS, poor mucociliary clearance, you add ETI, it's definitely rescued. And I think other people have shown that before. But when we had CF cells pre-treated with ETI and then added PFH, we didn't see that. And so this might indicate that our patients that have Pseudomonas and PFH may have a differential response to ETI. So how does this translate to an intact airway? So we then moved to using ex vivo piglet tracheas. And this was all work done by Namsu Ju. And we basically harvest the piglet tracheas and then put them in a bath of formoterol and carbachol, which really revs up mucociliary clearance. He has a poster kind of on these methods and his way of doing this. And then we either added phage or added PPS, and then this time it's ink toner on top, and we watch it move across. And here is a video of that. They should be synced, playing, yeah. So you can see the control where there's just PBS added on the left, and then on the right, the PF treated. And the PF is moving quite a bit slower. I think it's a little bit harder to see than the other videos. When we quantify that, and we did this in 2 separate experiments with 2 different tracheas in each condition, you can see the PBS treated in blue moves a lot faster for the full 30 minutes that we watched than does the PF treated. So in summary, phage can create liquid crystal and entangle cilia on epithelial cell cultures. Mucociliary clearance is impaired in epithelial cell cultures treated with PF phage, and then mucociliary clearance velocity is impaired in piglet tracheas treated with PF phage. So all things pointing to PF phage likely playing a role in the pathogenesis of chronic Pseudomonas infection in CF. And this will be on poster 52 later today. I also have a poster on the longitudinal cohort and the clinical associations. That'll be at poster 94. And again, like to thank my mentors and collaborators at Stanford, the Stanford CF team under Carlos Mila and the Boyke Lab, my funding sources, my new team at CHLA, and then my family, including my 2 family members with CF, who continue to inspire me. And with that, I'll take questions.
Hi, I'm Paula Zamora from Dartmouth. Those were very cool videos, by the way.
Yeah, it's fun
So
to
I
present.
have, I know this is not a phage therapy talk,
Yeah.
but I kind of have a
Yeah.
phage therapy tangential question.
Yeah.
So there are some papers that they have suggested the use of like aggregated phages as a way to kind of like preserve phage integrity during therapy. So with the idea that if you gave like an aggregate of phages, the phages that are inside are gonna remain viable. Do you think that the effect of the PFH on like disrupting cilia, it's like specific to a PFH, or it's like anything that you can give that it forms some sort of like macrostructure, and therefore, if this was like general, maybe we shouldn't be giving phages that aggregate in therapy?
I don't think so. We did a couple control phages. DMS is a lytic phage. FD phage is a E. coli filamentous phage. It's very similar to PF. And I think it's probably specific to the filamentous phages and their structure and the charge and the way they interact with things. I don't think the ones that aren't filamentous are going to have as much of an effect. But we did a little bit of work with this, so you can see there is some differences. But the PF was definitely more prominent. Yeah.
Hi, this is Danuta Radio from McGill University. I have 2 questions. The first one is how frequent this particular phage secretion is among different Pseudomonas strains, and whether it correlates with the different virulence, because we've seen a huge difference in response of epithelial cells to PA01, but that is extremely low virulent as opposed to PA14 or PA508.
Yeah.
So I was wondering to which extent you could draw some correlation between the expressions. So that's the first question.
Yeah, so PA1 carries PF4, so it carries one of the a filamentous phage. But as far as clinical isolates, so in CF patients' sputum with chronic Pseudomonas, at least in one cohort, I detected about a 40% rate of PF. But when I look at isolates, I actually detect the sequences a lot more. Like in the isolates I have from Stanford, it's more about 70%. So I'm still trying to figure out like if some of them don't make it, some of them do, or is sputum inhibition of the PCR just preventing me from detecting it? I don't know. And your other question was about the virulence.
Yes. So the correlation between the virulence, if the Pseudomonas strain is more virulent when it secretes and more of this phage? Or is there once the bacteria, once the Pseudomonas is infected, do they show, depend on the strain of Pseudomonas, similar secretion?
Yeah.
Phage,
I mean,
or?
so I'm still trying to figure out which ones exactly produce it more and those kind of things. But as far as viral, I mean, we think that it promotes chronic infection. It kind of protects the Pseudomonas. So, makes this kind of
so that actually
liquid
links
crystal.
to my second question.
Yeah.
What kind of effect you actually see? Because what is very nice about your work is that you were able to crystallize, so you have something that you can actually assess how these particular molecules are inflicting insult on the cells. And I was wondering whether you actually have looked at some inflammatory markers, at the change of the CFTR expression in these cells.
Yeah.
Even in wild-type cells, can they dismantle processing of the proteins? And
I haven't looked at CFTR expression. We did look at some cytokines in sputum. If my other poster, 94, if you want to come take a look at that later, I can walk you through it. But I did look at cytokines in sputum, and there is some pro-inflammatory and there's some antiviral signals, which is consistent. The antiviral is consistent with the mouse. Like there's the lab that I came from at Stanford did a lot with mouse models and wounds in this phage, and you definitely see an antiviral response.
So
more so than a
antiviral
bacterial.
response actually, because it will be interferon, so it actually will enhance to some extent also antibacterial, inflammatory.
Yeah.
Thank you very much.
Thank you.
Thank you. Very nice work.
Thank you.
Great work. This is Shu Wei Zheng from University of Washington. I'm wondering how long can those phages persist in the tissue? Will it actually be cleared or degraded by host or bacteria?
Yeah, that's a good question. I don't know that I can answer that. I mean, considering how when we have a patient who has phage in their sputum, we tend to detect it every sample. And then from the sequencing of the Pseudomonas that we've done so far, like patients don't tend to lose or gain phage within an isolate. It's like if whatever strain they have that has phage, it stays there. If they lose phage in their sputum, it's because they lost a strain of Pseudomonas, not because the phage like hopped out. So I think it's pretty persistent, but that's as much as I can answer.
Thank you.
Great talk, Liz. Jonathan Moff, Richmond, Virginia, VCU. Do you think the effect of phage on impairing mucus clearance is due to, just topology of cilia and ciliary function versus the phage affecting the mucus properties like viscoelasticity?
Yeah, I think it's more the mucus properties. It didn't show it, but we did measure ciliary beat frequency as well, and it was no different in those cultures. So the cilia are still beaten. It's just
Is your sort of setup allow you to do microparticle tracking to measure microbiology and stuff like that?
we're trying to figure out the microbiology on top of it. We tried to do microbiology of sputum that was infected with a phage, but it was just all over the place.
Yeah, but mucus itself, you might be able to do it.
Yeah, so that's one of the things that I think Carlos Mila at Stanford's going to continue to work on.
Hi, Liz. Great talk. Katie Heiser from National Jewish Health. So kind of building on that, this idea that your modulator data is really concerning.
I know.
And so in terms of, I know a little bit about this from Pat Secor's work and Laura Jennings' work, but, you mentioned it can aggregate with other things. And so, what can we do to try to break up, for people who already have phage?
Yeah.
Will pulmozyme help by trying to break up DNA, or is the phage just going to block that? And what do we have?
Yeah, I think the charge may be the key. It's something with the charge and small molecules and whatnot. And yeah, we haven't done a whole lot with it. We've talked about what things we could do for it. I also don't know with that data. I mean, that's those cells were pre-treated with 24 hours of ETI, so there's probably still mucus around that was produced prior to the ETI. So I don't know. We haven't followed up all of that, but yeah, I know. Like I said, we weren't really expecting that result. It was pretty dramatic. Thank you.
Great. Thank you very much.
Are you still nervous?
Yeah. That'll be good.
Yeah, it's better.
Keep me on my toes. right, my name is Jody Corley. I'm a postdoctoral research fellow in Dr. Katie Heizert's lab at National Jewish Health, and today I'm going to be talking to you about how antibiotics used to treat common cystic fibrosis pathogens influence its susceptibility to Mycobacterium abscessus infections. So I'd first like to thank the organizers for the invitation to present today. This is a great opportunity. Here are my disclosures. All right, so non-tuberculous mycobacteria, or NTM, pulmonary infections are a problem for people with cystic fibrosis, and these infections will likely remain a problem even though the introduction of CFTR modulators has greatly benefited many people with cystic fibrosis. This is because CFTR modulators do not resolve already established bronchiectasis or all infections with CF pathogens. Furthermore, NTM infections are increasing in prevalence globally, and even though NTM infections occur only in about 10 to 20% of people with cystic fibrosis, they're really important to consider because they are associated with poor clinical outcomes and are difficult to treat. So this brings up an important question, and that is, why do some people with cystic fibrosis get infections with NTM? And in answering this question, we can hopefully prevent future infections by predicting who is at risk, and then potentially determine better treatments for resistant infections. So in CF, there are two important types of NTM. They are Mycobacterium abscessus complex, or M. abscessus, and then Mycobacterium avium complex. My work really focuses on M. abscessus, so all of the studies you see today will be focusing with that organism. So what are the factors associated with M. abscessus infections? Bronchiectasis and Pseudomonas co-infection are associated with M. abscessus infection, but out of hundreds of possible mechanisms of susceptibility, some important factors to consider would be structural damage of the lungs, direct interaction between Pseudomonas and M. abscessus, and secreted products from Pseudomonas. What I'm interested in is a potentially modifiable mechanism, and that is which involves understanding what the effects of antibiotics used to treat other cystic fibrosis pathogens are on M. abscessus, specifically antibiotics used to treat Pseudomonas aeruginosa. So most people with CF have Pseudomonas infections, and these infections are continuously treated with antibiotics that are generally not considered effective in the context of M. abscessus, as they don't kill M. abscessus. However, I would like to know if they change M. abscessus in some way to promote infection in the host. So to begin to answer this question, I've been looking into a transcription factor known as YB7, whose expression is induced by antibiotics. But the identified antibiotics that are known to induce its expression are antibiotics that are used to treat M. abscessus itself. So YB7 is actually conserved in actinomycetes and has been primarily studied in M. tuberculosis and Streptomyces species. But looking at part of the YB7 regulon in M. abscessus, other groups have found that it regulates genes that promote resistance to antibiotics, oxidative stress, and phagocytosis survival. So the regulon in YB7 itself has been primarily studied in the context of antibiotic resistance, but I'm interested in understanding how YB7 may promote M. abscessus infection. And so to get started here, we have some data from a group demonstrating that subinhibitory concentrations of amikacin, which is an antibiotic used to treat M. abscessus, actually induce expression of YB7, as shown by QRT-PCR here. Furthermore, the same group saw that treating M. abscessus with subinhibitory concentrations of amikacin also promoted M. abscessus growth in THP1 cells, which are a monocyte cell line. And they did this by measuring colony-forming units over a time course. They also saw that if they overexpressed YB7 on a plasmid, that they saw a similar effect in growth. They saw increased growth of M. abscessus in the THP1 cells compared to the vector control strain. So these studies were looking at amikacin, but I really want to know what other antibiotics specifically not used to treat M. abscessus do to promote YB7 expression and M. abscessus growth in the host. So what about the antibiotic tobramycin, which is used to kill Pseudomonas? Does this antibiotic actually induce YB7 expression? So I've shown you that amikacin, which is used to kill M. abscessus, induces YB7 expression. Tobramycin, on the other hand, is actually used to kill Pseudomonas and is actually given frequently to people with cystic fibrosis on a continuous basis. Since most people with CF have or had PA infections or Pseudomonas infections, tobramycin is often used as maintenance therapy or as treatment for exacerbations, which is one of the reasons why I wanted to look at its ability to induce YB7. Additionally, it's very similar to amikacin in the sense that it's also an aminoglycoside antibiotic, which has a similar mechanism of action. So, given this information, I really want to ask the question, does tobramycin induce YB7 expression? So my hypothesis is that, yes, tobramycin used to treat Pseudomonas and CF airways will enhance M. abscessus growth in vivo by inducing expression of YB7. So to begin with, I wanted to check to see if tobramycin actually does induce YB7 expression in M. abscessus. So I began by pre-treating M. abscessus with amikacin or tobramycin, extracted RNA from the bacteria, and then performed QRT-PCR. So what you can see here is that M. abscessus YB7 expression was increased after treatment with amikacin, and interestingly, it was actually increased with treatment of tobramycin. So we do want to evaluate this information to see if this is a dose-dependent effect. So we're currently evaluating dose and exposure time of tobramycin treatment. And in the future, we do want to test other antibiotics to see antibiotics used to treat Pseudomonas or staph infections like Bactrim and levofloxacin to see if they have a similar effect. So next, I'm in the process of evaluating whether or not aminoglycoside pretreatment of M. abscessus promotes the growth of M. abscessus in a primary cell line. So previously I showed you data that demonstrates that M. abscessus grows more when pretreated with amikacin in a THP1 cell line, but I want to really check the clinical relevance of that. So after isolating monocytes from healthy donors, we mature them to macrophages, and then we infect them with M. abscessus incubated with, or pretreated with amikacin for an hour, and then we infected the cells and then quantified CFUs over 96 hours. So what we can see here is that with amikacin pretreatment, we get an increased growth of M. abscessus compared to untreated bacteria. So this suggests that amikacin does indeed promote M. abscessus growth in primary macrophages, similar to THP1 cells. So the next steps that are in progress currently are to determine if tobramycin promotes M. abscessus growth in macrophages too. So next, I wanted to see if aminoglycoside treatment improved the ability of M. abscessus to resist oxidative stress induced by reactive oxygen species, or ROS. So to do this, I performed assays to determine the minimum inhibitory concentration of hydrogen peroxide. What that minimum MIC was to inhibit growth of M. abscessus using M. abscessus pretreated with either amikacin or tobramycin and then exposed to varying concentrations of hydrogen peroxide. So what we can see here is that compared to no antibiotic treatment, both amikacin and tobramycin actually increase the MIC, or the resistance to hydrogen peroxide, suggesting that antibiotics may help the bacteria survive or be better adapted to oxidative stress. Oops. So to kind of sum up the experiments I've done so far, I've shown you that aminoglycosides like tobramycin induce YB7 expression, and that amikacin promotes growth in human primary monocyte-derived macrophages. So this tells us that antibiotics promote M. abscessus growth and YB7 expression, but I want to see the relevance to YB7 itself. So I next wanted to ask, what is the role of YB7 in this pathway? Does this activity depend upon YB7? So I asked the question, does knocking out YB7 impact M. abscessus growth? So looking at both human primary MDMs and primary human alveolar macrophage infections, when we infect with the M. abscessus YB7 knockout strain, we do see fewer CFUs in these MDMs or alveolar macrophages compared to the wild-type M. abscessus strain. So this does suggest that YB7 is important for growth in primary human macrophages. Additionally, we wanted to see if the M. abscessus YB7 mutant was more susceptible to reactive oxygen species than wild type. So I performed a hydrogen peroxide MIC similar to what I just showed you. And what we can see here is that the YB7 knockout is indeed impaired for ROS survival. So the next step would be to pre-treat the M. abscessus YB7 knockout strain with tobramycin and amikacin to see if we still see an impairment in ROS survival. And I anticipate we will, since we believe that the increased hydrogen peroxide resistance seen with antibiotic treatment is the result of YB7 activation. So taking these data and the human macrophage infection data together, we can see that YB7 promotes resistance and survival. And to sum up everything, aminoglycosides like amikacin, which is used to treat M. abscessus, and importantly, tobramycin, which is used to treat Pseudomonas, induce YB7 expression. They also promote resistance to reactive oxygen species, and amikacin itself promotes growth in human primary MDMs. And again, I am repeating this experiment after exposing the bacteria to tobramycin. If we knock out YB7 and M. abscessus, M. abscessus does have a growth impairment in both human MDMs and alveolar macrophages. But I really would like to understand, or to prove that this is actually going through YB7. So I have a few pending experiments that are currently in process. The first would be to assess the dose and time course for optimal YB7 expression, and then assess the requirement of YB7 for persistent infections using an acute marine model of M. abscessus infection. Then determine if the administration of tobramycin impairs marine lung clearance of M. abscessus. Then we're also working on RNA-seq studies to determine the YB7 transcriptome in the context of tobramycin treatment and intracellular infections. So this is really important to consider, because understanding what the role of both YB7 and antibiotics used to treat other CF pathogens play in promoting M. abscessus infection may help predict who's susceptible to M. abscessus infections and then inform future treatment strategies. So I'd like to thank my lab, who've been very supportive. Alma and Jack have helped with some of these experiments. Everybody at National Jewish, my CFF postdoctoral fellowship mentoring committee, and then Kyle Road for providing the YB7 mutant strain. I'll take any questions.
Hey, Katie, this is a great talk for Restart Myth. So I was wondering if you have a sense if... Oh, jeez.
Watch out, man. It's popular.
Oh, it's mine. Oh, wow.
Yeah.
Do you know if there are like any clinical strains of M. abscessum that have gain of function in YB7?
So I don't know that for sure, but we have screened isolates. Well, I haven't screened them, but we dug into the data of some M. abscessus isolates that looking for YB7 mutations. We didn't find any YB7 gain of function mutants, but we did only find one synonymous mutation in YB7 out of hundreds of isolates, suggesting that it is actually important probably for infection. So that's at least one answer. So
It was a great talk. Thank you very much. Valerie Waters from State Kids in Toronto. I'm interested in your choice of concentration of aminoglycosides. Because years ago, Lou Kaufman showed that sub-inhibitory levels of tobramycin increase biofilm growth of Pseudomonas. 1 microgram per mil is really low.
Right.
It's below the Clisey breakpoints, and with inhaled, you get on mean 1,000 micrograms per mil. So would you consider repeating them using concentrations that we actually see in the airways with inhaled drugs? Because I wonder if you might get a different result.
Yeah, that's one question we're really considering, and the time course that I'm working on will actually hopefully address that. But yeah, definitely a good consideration.
Hi, Jody. That was great.
Thank you.
From the University of Washington. I'm wondering if you have considered other factors than YB7 and whether or not you would, it's quite possible that this is all mediated the same way it is by amikacin, but you could also just perform an RNA-seq on tobramycin versus amikacin and see if you elucidate other possible pathways.
That's a good point.
That might mediate this effect.
Yeah, we can consider that. It is possible, and hopefully, RNA-seq studies will kind of extrapolate that. YB7 is a global regulator, so it does regulate a whole bunch of different processes. So I'm assuming even if it is a different pathway, it probably ties in together with YB7 somehow. So.
Hey, Jody, great talk. I was worried about Ajay stealing my question. So.
So I was curious too about the MIC and if it was sub-MIC, because I don't know for mycobacterium, but for other bacteria, when you treat with sub-MIC levels, it can push them towards biofilm. And so I was wondering, I know M. abscessus is clumpy, right? So I didn't know if you saw with your pretreatments, did you see
Oh, yes.
differences in like
Oh.
aggregation or biofilm
Yes.
formation? Is that something that could also be playing into the either the survival or the phenotype within macrophages?
That's definitely a huge consideration because when we do pre-treat with any antibiotic, it does become extremely clumpy. So yes, that could be playing a role. Yeah, it does. It does seem to help modify the membrane somehow to become more clumpy, even with the glycolipopeptide still intact. But yeah, that's definitely something we need to consider in our assays.
I have a question. I have two questions. So we see more and more amikacin-resistant abscessus. Have you looked at any resistant strains with maybe even higher levels of?
I have not, but that's a great idea. That's a great idea. I have not. Yeah, thanks.
And with RNA-seq that Katie's going to pay for.
Yeah.
And then the other thing was, is it known what the real proximate signal is for YB7? Like what's actually turning it on?
I don't believe it has. I mean, there's several studies looking at the effects of oxidative stress on changing something in the promoter region. But other than knowing that antibiotics or oxidative stress induce its expression, I don't think much is known as what exactly the signal is that activates it.
Cool.
I just want to make a comment that YB7 is a great name for something you're trying to understand, like what it does. Like, YB7, I think that's such a good name.
That's what our lab researcher said too. He says that all the time.
I do want to make. I know we're about to wrap up. There are questions in the text and comment. I apologize if I didn't get to all of them. Most of them I think have been answered. So from all the talks, this doesn't refresh very well. So I apologize. But if there's anything I missed, please call me out, text into here, and I'm happy to announce it. But I think most of these have been answered. So.
Great.
Thank you.
Yeah. Thank you.